In the previous video, we discussed what stem cells are, their uses, terms like potency and self-renewal, as well as the four main types of stem cells and their specialization. In this video, we're going to talk about how we derive stem cells and compare their differences. Let's start by talking about the sources of stem cells. So first we have the embryonic stem cells. These are derived from a very early stage in human development, and they have the potential to produce most of the body cell types. Second, we have the adult stem cells. These are found in certain tissues such as the bone marrow and fully grown humans, and they can be limited to producing only certain types of specialized cells. Third, we have the umbilical cord blood and the placenta. They can give rise to many stem cells, mainly the hematopoietic stem cells and the mesenchymal stem cells. The hematopoietic stem cells form blood, and the mesenchymal stem cells form body tissues. Let's discuss the embryonic stem cells. Embryonic stem cells are gotten from the inner cell mass of the blastocyst. The blastocyst, of course, is the pre-implantation embryo that develops just 5 days after fertilization of eggs by sperm. So these are the advantages of embryonic stem cells. Firstly, they are flexible because they can produce almost every type of cell in the body. And secondly, they are easier to collect, purify, and maintain in labs compared to adult stem cells. The disadvantages are, of course, the first one would be the ethical concerns regarding the destruction of an embryo that could have resulted in a baby, and that they can replicate in an undifferentiated state for a long time before they're stimulated to create specialized cells. Now, the problem with the undifferentiated stem cells is that we can never use them for tissue transplants because they can cause a tumor called a teratoma. Now we're going to discuss the sources of embryonic stem cells. So first we have in vitro fertilization. As we've said, this has raised ethical issues with using embryos since the blastocyst will never develop into a human. A solution for this would be to donate the blastocyst for adoption by infertile couples. Secondly, we have something called the somatic cell nuclear transfer, or therapeutic cloning. In this method, we insert the nucleus of an already differentiated adult cell, like a skin cell, into a donated egg that has had its nucleus removed. So here we have a regular adult cell and its nucleus. And here we have an egg cell without a nucleus. So we're basically just going to take the nucleus here and transfer it to this egg cell. The egg with the genetic material of this cell is then stimulated to form a blastocyst from which embryonic stem cells can be derived. The stem cells created this way are clones of the original adult cell because their nuclear DNA matches the adult cell. This, however, has yet to work with humans. Now let's compare the sources of stem cells. Here we have the embryonic stem cells, the adult stem cells, and the cord blood stem cells. We're going to talk about their attributes, their limitations, and ethical concerns. Let's start with the embryonic stem cells. We have the IVF and the nuclear transfer. The IVF, of course, is the in vitro fertilization. So with IVF, we can produce all cell types, same with the nuclear transfer. It is very relatively easy to identify, isolate, and maintain and grow them in a lab, same with nuclear transfer. And with the IVF, there's a large source of excess blastocysts from IVF clinics. In the nuclear transfer, the stem cells can be genetically matched to the patient. This lowers the chance of the patient's body refusing the treatment. The limitations here are with IVF, there is a risk of creating teratomas if we do end up using the undifferentiated stem cells before they become specialized. And with the nuclear transfer, it has yet to be done with human cells, and there's also the same risk of creating teratoma from the undifferentiated stem cells. Regarding the ethical concerns with IVF, you do end up destroying the human blastocysts, and same with the nuclear transfer, but also with the nuclear transfer, there are fears of misapplying this technique and reproducing actual clones of adult tissues. Regarding the adult stem cells, they have shown success in some treatments and they can be genetically matched to the patient. The limitations are that they produce limited amounts of cell types in comparison with the embryonic stem cells. They're not found in all tissues and they are difficult to identify, isolate, maintain, and grow in the laboratory. So far, there haven't been any ethical concerns raised yet. With the cord blood stem cells, they are more flexible than the adult stem cells in terms of differentiation. Less mismatch problems happen in bone marrow transplant, and there are no graft versus host rejections, so the body does not attack them. The biggest limitation with cord blood stem cells is that the stem cell count, which is done after the separation, is sometimes lower than the amount that can be used directly. Ethically speaking, there are no ethical issues that have been raised. I hope you were able to learn about how we derive stem cells and the differences between their sources. Please consider subscribing to this channel for more videos of this nature. Thank you for watching.